Okay, so let's begin. So this is the general lecture on bioenergetics. So this first lecture was designed to be super simple to understand and something that doesn't take a lot of effort for a beginner in any field to be able to make sense of. So what I'm going to basically do in this lecture series is that first I'm going to introduce the concept of bioenergetics, what are the different areas you can use bioenergetics in biology, like where all it is useful. And then I'm going to talk about how we study bioenergetics, what are the different factors that influence bioenergetics as a whole. So to begin with, what we think of bioenergetics is that it is the let me just get my pointer. Yeah, it is the breakdown of a large carbon chain. So they can be in the form of carbohydrates, fatty acids, or proteins. Breakdown of these long chains of carbon into shorter carbon chains. And in the process of breaking down into shorter carbon chains, what we know is that it usually produces a lot of ATP. And that's what we think of bioenergetics when we talk about bioenergetics. When I tell you, okay, this is bioenergetics, you think of ATP production, ATP utilization, and so on. But bioenergetics is not just ATP production. You can also have the generation of reducing equivalents. So these reducing equivalents that I'm talking about are NADH, FADH, FADH2, and so on, NADPH even. So these reducing equivalents can either influence anabolic reactions or they can go on to produce ATP themselves. So not produce ATP, lead to the production of ATP through the electron transport chain. And you can also have biomass generation. And this biomass generation is usually in the form of anabolism. So these carbon chains that are broken down are not always completely broken down to carbon dioxide. You can have intermediate carbon chain lengths, and these can then be used for the generation of biomass. So overall, bioenergetics can be utilized in various processes, which is to maintain the redox homeostasis of the cell, to manage the waste inside a cell. You can have the maintenance of calcium homeostasis. So calcium is super important for muscle contraction and various other functions. So the management of calcium inside a cell is also mediated by the mitochondria and bioenergetics. One of the part is mitochondria. It's not just mitochondria. You can have the regulation of the function of certain proteins. So you have a lot of proteins that depend upon the presence or absence of ATP. You have NAD dependent proteins, so they have their catalytic activities are dependent on NAD. And you can also have the regulation of gene expression. So the metabolites that I spoke about, you can have different amounts of metabolite in a cell based on various bioenergetic processes, which can influence the epigenetic landscape of a cell. And if you influence the epigenetic landscape, you therefore influence gene expression. And finally, biosynthesis. So, like I spoke about the generation of biomass, you have biosynthesis, which is very largely not like it's deeply connected to the overall concept of bioenergetics. Yeah. So when I largely talk about bioenergetics, I'm mostly referring to the generation of ATP, like I spoke about earlier. So right now we're going to focus on the right now we're going to focus on the generation of ATP in various double membranes, or not the generation of ATP. It can just be how energy is transduced in a biological system. So, oops, yeah. So our foremost example is the mitochondrial inner membrane. So you basically have two membranes in the mitochondria. You have the inner membrane and then you have the outer membrane. So the solid line is the inner membrane, the dotted line outside is the outer membrane, and you have a positive and a negative side. So the intermembrane space, the space between the two membranes is filled with protons. That's what we know as the proton gradient. And this is the matrix of the mitochondria. So this is where proton is being pumped out from. So naturally, protons have proton which is H plus, H plus. So if you have pumping of protons happening in one direction, that side is going to become positive. So this is the intermembrane space which has become positive, and this is the matrix which, in relatively to the intermembrane space, is negative because it has the protons have been pumped out of here. So what happens basically is you can have the pumping of protons through various energy inputs. So the energy input for say a regular mitochondria or say a bacteria that is breaking down say carbohydrates or fatty acids without utilizing light, that can be through electrons. And this flow of electrons is what gives the proton pumps the energy to pump protons out. So therefore, these proton pumps are dependent upon the electron transport chain, and this electron transport chain is the same that we have inside the mitochondria. So you have this electron transport chain in the mitochondria, which we are going to talk about like in depth later on. So basically, this transport of electrons in the mitochondria is what is giving you the ability, the proton pump, the ability to pump protons out. So here in this first case, it's through the pumping of electrons, sorry, the flow of electrons. And in the second case, say you have photosynthetic bacteria, photosynthetic the plants. So you have chloroplasts in these plants. What here is basically happening is that they're using photons as a primary energy source. And by utilizing photons as a primary energy source, what they're doing is that they're pumping protons in one direction. So instead of using electrons in this case, you can also use photons as a primary energy input. So here I've just basically introduced the concept of a primary energy input. You can have either an electron flow or you can have a photon directly donating the energy. And uh, it, largely in this lecture and the next few lectures, what I'm going to focus on is the electron flow. And this electron flow through the electron transport chain, or in the mitochondria at least, is what we're going to largely focus on. And then eventually later on, uh, if time permits and if I have the time to design a lecture on the photosynthetic bacteria and plants, we'll do that. Okay, so so this is the mitochondria. So this is basically just an electron microscope image of the mitochondria. This has been given false color. Basically, the magenta pink membrane that you see. So that's the mitochondrial membrane. The outer surface of it is the outer membrane. The inner surface of it is the inner membrane. The invaginations that you are seeing into the mitochondria. So this blue part is the inside of the mitochondria, and these invaginations are the cristae. So you have basically four, five important markers to remember in a mitochondria, which is the outer membrane, which is the outer layer here, the intermembrane space, which is the space between the two inner and outer membranes. Then you have the inner membrane, the cristae, which is an extension of the inner membrane, but the cristae and the inner membrane are quite different from each other because they differ functionally, and not all cristae are equal to simply the inner membrane. So you basically have different protein profiles that are embedded into the crystal membrane versus the inner membrane, but we will get into what is the cristae and stuff in future lectures. And basically the matrix, which is the inside of the mitochondria, here is where you have all the biochemical magic happening, where you have accumulation of metabolites, the regulation of transport of metabolites, and basically everything that you need to have this electron transport and ATP generation. That is all, the metabolites are all inside the matrix. So again, I'll just pause for a second to see if there are any questions. Okay, so what is the dotted line on the showing mitochondrial bacteria? Okay, I'll just go back to that slide. Yeah, on this slide, is that what you were asking about? Okay, so the dotted line here represents the outer membrane. So basically, you need two membranes to maintain this proton gradient. The dotted line is the outer membrane. The solid line is the inner membrane. Then does the one showing bacteria only refer to gram negative? So I'm not talking about the cell wall here. And in bacteria, you can have the periplasmic space, which can also serve as somewhat of like a block for the proton. So not always. So it's not always true in the bacteria, but yes, if you have very clear two distinct walls, then yes. Yeah, so we're not going to focus on bacteria at all for the first few lectures at least. Here I'm going to focus on the mitochondria largely. In any case, so yeah, I spoke about the mitochondria here. So, like I was talking about the electron transport chain and how the ATP is being produced. So, I'll just go to very briefly give an overview of how you have the transport of electrons into the electron transport chain in a mammalian mitochondria. So, basically, what you have here is you have different substrates. You have glucose, you have fatty acids, you have amino acids and proteins. So we know these are the large macronutrients that we talk about that the body is burning. And the burning of these, so the burning, double quotations, is what we colloquially refer to as basically breaking it down into smaller carbon chains, like I spoke about earlier. And this breaking down of smaller carbon chains eventually, for almost all substrates that you utilize, which is basically the fuel of the body, eventually leads into the tricarboxylic acid cycle. So the TCA cycle or the Krebs cycle, this is inside the mitochondria. And here what you basically have is a generation of reducing equivalents. You can have the generation of reducing equivalents in other parts of the in other like other processes as well. But here I'm just going to talk about the TCA inside the mitochondria and NADH and FADH2. These are the two reducing equivalents that I want to talk about that are here. And NADH donates electrons to complex 1, an electron pair, one pair of electrons for one NADH, and an FADH2 donates a pair of electrons to complex 2. So you have these five complexes in the mammalian electron transport chain 1, 2, 3, 4, and 5. You also have the coenzyme Q and cytochrome C that are shuttling electrons between complexes. And this is basically your electron transport pathway. You can have the electrons being donated at complex 1. It can then be transferred to the coenzyme Q, which gives it to complex 3. Complex 3 gives it to cytochrome C. Cytochrome C then gives it to complex 4. At complex 4, these electrons are involved in the generation, in the consumption of oxygen. So when we breathe oxygen, a large part of the oxygen in our body is utilized to support this electron accepting feature. So the final electron acceptor in our body is oxygen, and that's why we largely breathe oxygen to serve as the final electron acceptor at complex 4. So you have this electron finally being accepted into oxygen, it forms water. And then in this entire process, what's happening is that most of these complexes, so this I'll just zoom into the electron transport chain, complex 1, complex 3, and complex 4. These complexes are involved in shuttling protons across the membrane. So I talked about proton pumps, right? They utilize the energy of the flow of electrons to pump protons. 1, 3, and 4 pump protons across the inner membrane. And this is these protons are accumulated in the intermembrane space, and this gradient of protons, you have more protons in the IMS, less protons in the matrix, so you basically have a gradient. This gradient of protons is utilized by the ATP synthase, which is complex 5. ATP synthase uses these protons, so it's basically like a it's basically like you have a water tank and you have a tap at the bottom of the water tank. And if you turn the tap on, the water starts flowing through the tap. And ATP synthesis is basically doing that. It's basically allowing protons to pass through it. And in allowing protons to pass through it, it is generating ATP. So it's basically, it's literally a motor where it's rotating uh, because of the proton flowing through it. And this rotation is causing ADP and an inorganic phosphate to combine and give you adenosine triphosphate, or ATP, which we colloquially refer to it as the um, energy currency of the cell. But this this proton gradient that I talked about, the only way it can be dissipated is not through the ATP synthase. What you can also have are other proteins which can utilize this proton gradient. And a lot of these proteins are referred to as uncoupling proteins. So the uncoupling proteins that I've shown here at the end, you have uncoupling proteins which can utilize the proton gradient for it to fall back into the matrix. And these proteins are not always involved in the generation of ATP. You have the ATP synthase, which uses the protons to make ATP. This is known as coupled respiration. So that's what we want to happen. Like, we want a lot of ATP, we want a lot of coupled respiration to happen. And if it is transferred through other proteins or other ways, you can have proton leak through various ways. It is known as uncoupled respiration because the dissipation of the proton gradient is now not linked to the generation of ATP. This is known as uncoupled respiration. So you basically have coupled respiration, which is the dissipation through ATP synthase, and uncoupled respiration, which is basically any way where you can have a leak of protons through the membrane. So sometimes the protons can be leaked through some specific proteins. Sometimes fatty acids are also involved in shuttling protons. You also have proton leaks directly through the membrane. And so there are various ways you can have a proton leak in your system. And as you can imagine, with a leak, if your water tank is leaking, not all of your water is being utilized correctly. Therefore, sometimes uncoupled respiration is also referred to as wasteful respiration. But as I go along in future lectures, we will see why uncoupled respiration is not wasteful at all. There are various ways you can have where uncoupled respiration can be utilized in a cell to give you various useful things. So yeah, so I was talking about the proton gradient. The proton gradient can regulate thermogenesis. So thermogenesis is basically heat generation in the body. So your brown adipose tissue, your skeletal muscle, what happens largely is you start shivering when you're cold, right? But uh, there is also a kind of thermogenesis which is known as non-shivering thermogenesis. And non-shivering thermogenesis, what you have is that the proton gradient is passed through an uncoupler, and the energy that is released through the flow of the protons is 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 released as heat, and that's what is used to heat the body up. So this is known as non-shivering thermogenesis. So the one useful feature of uncoupled respiration, you can have metabolite transport, which is affected by the proton gradient. So here I'm just talking about pyruvate. So all of these data are just to support my point. I will go and talk about each of the graphs that I have put on the screen. I'm going to talk about individually later in the lectures. This is just to give you a taste of what kind of things I'm going to talk about. You can have changes in calcium homeostasis dependent upon the proton gradient. ATP ADP transport. So we know ATP is produced inside the mitochondria. Have you thought about how is this ATP inside the mitochondria now going out into the cell? Because the cell also requires this ATP. It's not just the mitochondria that requires ATP. So the transport of ATP ADP is also a very interesting part of is also dependent on bioenergetic regulation. It's quite interesting to study about. You can have ROS production, so reactive oxygen species, which are uh, which are basically damaging to the cell. These are oxygen radicals or other reactive oxygen species which can damage the cell in various ways. And finally, autophagy. Autophagy is basically where the cell is breaking down some components of it, either as a protective mechanism or when it doesn't require certain organelles or proteins anymore. So autophagy is also dependent upon the proton gradient itself. So these are just some processes that are dependent upon the function and the amount, the dissipation of the proton gradient. Yeah, so I was talking about the proton gradient for a long time, but how do we measure the proton gradient? Like, how do we make sense of this entire thing? So basically, you have the proton motive force. This proton motive force is basically the energy stored as the proton gradient. This is just colloquially I'm explaining. We talked about the proton gradient, and I briefly explained what the PMF is. So how do we measure the PMF? How do we make sense of this entire thing? So the PMF, or the proton gradient, can be regulated at various levels. And this regulation can be at the level of generation of this gradient. You can have a choice between where do you want your electron to enter the system. Because if you notice carefully, complex 1, 3, and 4 are the ones that are pumping protons. 1 pumps about 4 protons, 3 pumps about 4 protons, and complex 4 pumps 2 protons. But if you have a FADH2 donating electrons at the level of complex 2, you are skipping out on the 4 protons that are pumped as a result of complex 1 activity. So it makes a difference whether you have NADH that is donating electrons or FADH2 that is donating electrons. Because if you have NADH that is donating electrons, you can have about 10 protons that are pumped. If you have FADH2 donating electrons, you can have about 6 protons that are pumped. And also the number that I'm saying, 10 and 6, that's not always uh, the case. You can have electron slips, proton leaks, as I spoke about. So it's not always a round number. You have 10 protons being pumped. It is close to that number. And I'm going to talk about what are these numbers a bit later. So the generation of this gradient at the level of complex 1 or complex 2, you can have the electron flow itself, which can be altered by complexation of the mitochondrial complexes. You don't have individual complexes that are acting alone, right? They need to transport electrons to the next complex in line. Therefore, sometimes they cluster up together, and when they are clustered up together, you would imagine that due to lower physical distance, it is much easier to transport or much faster to transport electrons in that manner. So super complex formation, where a lot of these complexes come together to form a super complex, is improves electron flow in many conditions. And so there are various factors that affect electron flow. And if you have deviations in the electron flow, you can imagine you will have deviations in the proton pumping itself. And you can have the dissipation of this gradient that is affecting the gradient itself. So I spoke about ATP synthase, which is the coupled respiration. This is dissipating the gradient, making ATP. And then you have uncouplers that are dissipating the gradient but not making ATP, but are doing other useful things in the cell, just not ATP production, therefore known as uncoupled respiration. So therefore, these are not all the ways you can control the proton gradient. There are many, many factors that will come into picture, but for now, I'm just going to stick to these. So to make sense of this proton gradient, we have these various ratios that give us an idea about how much ATP is generated per oxygen molecule consumed. So the PO ratio, so this is the first ratio here, the PO ratio gives you an idea about how much ATP is being generated for the amount of oxygen that is being consumed. You can have different amounts of ATP being generated for the same amount of oxygen that you are consuming in a cell. So this factor is going to depend upon the protons that are being pumped, the oxygen that is being provided to the system, the amount of ATP that is generated, which is going to depend upon again the amount of ADP in the system, the amount of inorganic phosphate in the system, and the protons themselves undergo various types of regulations. So sorry, I mean the proton gradient. Therefore, the PO ratio is going to change in different contexts. And what I was going to discuss next was the breakdown of this PO ratio, but I don't think I'll do that today. I'll probably do that in a future lecture. I'll discuss all of these subsidiary ratios later in a later lecture. Yeah, and this is just giving an idea of the various PO ratios you can have through complex 1 or complex 2 mediated substrates, but I'll come to substrate choice and what the body and the mitochondria is using later. I'll talk about this in a future lecture. So it is not just the proton gradient or the substrate that you're using. Is it complex 1? Is it complex 2? It's not just these factors that are going to affect your proton gradient and bioenergetics as a whole. You can have dynamics of the mitochondria itself. So the shape of the mitochondria is also going to dictate how efficient this mitochondria is at a certain task. So for sorry, you can have either fissioned mitochondria, which are fragmented mitochondria. You can have individual mitochondria that are spread across the cell, or you can also have a fused network of mitochondria, where a lot of these mitochondria are technically fused to each other to form a long tubular structure. And this fission and fusion of the mitochondria, this is what we refer to as mitochondrial dynamics. You can have either of these states in different energy conditions. When you have a low energy demand, um, you don't need the mitochondria to be very efficient. You have a lot of nutrients being supplied to you. In that case, what we have usually seen is that the mitochondria are fragmented. But when you have a very high energy demand, when you require a lot of energy, when the amount of substrates available are not very high, when you need a lot of efficiency from the mitochondria in terms of ATP generation, then we see a lot of these tubulated and hyperfused mitochondria coming into the picture. And this is just one paper that I've put here that is talking about the shape or the dynamics of the mitochondria in different conditions. You have untreated cells, you have cells treated with PBS or HBSS. PBS and HBSS are just two kinds of media that we can keep cells in. So PBS is not technically a media, but yeah. In any case, PBS and HBSS are both mimicking a starvation-like condition where you don't have nutrients provided to the cell. And when you don't have nutrients provided to the cell, what they are seeing is that so tubulated is the dark blackish, the dark gray bar. From untreated to a nutrient starvation condition, there's an increase in the tubulated mitochondria, and there's a subsequent decrease in intermediate and fragmented mitochondria. So that was just I just brought one example to highlight that this is actually happening in cells, and people have shown it happens um, in other model systems as well, not just cells. You can have the proteostasis of the mitochondria that is affecting bioenergetics. So proteostasis is basically uh, the number of complexes that are present. So you can imagine if I have a lot of complexes in the mitochondria, I'm going to have a lot more ATP production. And if I have a lot less complexes in the mitochondria, I'm going to have less ATP production. So the dynamics of the proteins themselves come into picture. You can also have the changing of the gross number of the mitochondria itself. So you can have an increase in mitochondria, which is mitochondrial biogenesis coming into picture. You can have decrease in mitochondrial numbers, which is mitophagy. You can have mitophagy come into the picture, which will change the amount of mitochondria naturally present inside a cell. So that's going to again affect how much ATP you're producing, how are you affecting bioenergetics, and therefore how are you affecting the cell. So I'm going to again talk about uh, these intrinsic mitochondrial factors in a separate lecture. So I'll have one other lecture just focused on the mitochondria and how the mitochondria is controlling bioenergetics. And finally, so this is my final slide. I'm going to stop after this. And here basically is just a representation of the total energy expenditure. So what does total energy expenditure mean? Total energy expenditure is, I'm not going to explain how experimentally they do it in the field, but what it basically gives you an idea about is how much is your body expending energy. How much energy is your body utilizing at any given time is given by a total daily energy expenditure. And what here it is shown is that, so the first graph is talking about fat-free mass. So fat-free mass is basically the mass of the body free of after removing the fats. The fat, as you see an increase in fat-free mass on the x-axis, you also see an increase in the daily energy expenditure, the total energy expenditure on the y-axis. And this seems true for various groups of humans. So they did this study in different age groups. I think they started from neonates up to very, very old people, 80, 90-year-old people. And as you can see, as you grow older as well, your so the age is shown on the x-axis, the energy expenditure is still on the y-axis. For both males and females, they were able to calculate the total energy expenditure for various groups of humans at different stages of their life. And what they can see is that as you grow older, there's a change in how much energy your body is utilizing. So there's a rapid increase in the energy expenditure as you're growing. So about till maybe 12, 13, 15-ish years is a very rapid change, and then it starts plateauing from about 15 to 20, and from mid 20s onwards you start to see slight dipping in the amount of energy your body is consuming. Basically, it just tells you that at different stages of your life you have different amounts of sorry, different amounts of energy that is being utilized by your body, and therefore it becomes important to know what kind of energy generating dissipating mechanisms exist in the body, and what how are they contributing to this daily energy expenditure, because you can have an increase in uncoupled respiration perhaps that's going to largely reduce ATP production by some degree, and that might increase your energy expenditure because you're using more fuel to maintain the same amount of ATP. What if you have a reduction in uncoupled respiration? In that case, you're going to start using less fuel to maintain the same amount of ATP. In that case, you might even start accumulating fats because you're not utilizing the food you're eating as much as you're supposed to. So, changing these mitochondrial factors can affect your daily energy expenditure, and at different stages of your life, it's going to affect you differently. And so I'm not going to go into this paper at all. I just wanted to bring into light the fact that energy expenditure is not just something that happens at a cellular level. We don't just study energy isolated to mitochondria or bacteria. We do this at a whole body level as well. And at a whole body level as well, we can capture differences at different stages of your life between the sexes themselves, um, when you're doing different kinds of activities. So, bioenergetics is not just, you know, how much ATP is being produced and ends at that. It's going to control cellular function, organismal function, how you interact with society, how well you're able to, how well equipped you are to deal with different stresses of life, and so on. So I just wanted to leave with this final thought that, yeah, bioenergetics is relevant. It's very important to know the details of the mechanisms that regulate the energy providing and utilizing processes inside a cell and inside your body. So yeah, that was the lecture for today.